Hello, everyone. My name is Sophie Hendricks, and I'm an assistant professor at Hasselt University in Belgium. And in today's lecture, I will give you an introduction to transcriptomics. So the goal of transcriptomics is to compare gene expression levels between different cell types or tissues within the same organism, or between healthy and diseased cells or tissues, and also between cells or tissues which were either or not exposed to stress conditions. And these stress conditions could, for example, be exposure to a certain toxic compound. So as you can see in this figure on the right, in our cells, DNA is transcribed into mRNA or messenger RNA, and this is then translated into a protein. And the protein will then carry out a biological function in a cell. And when we perform transcriptomics analysis, as the name implies, we are focusing on the level of the messenger RNA or the transcripts, as we also call them. So when you want to determine gene expression levels, you could use different methods. The ones that I've listed here are not the only methods that are available, but I think it's the most used ones. So these are real-time PCR. It's also called quantitative PCR or qPCR. We also have Northern blot, microarrays, and RNA sequencing. And the focus of this lecture series will be on real-time PCR, but I will also briefly introduce the other three methods to you today. So when deciding on which method to use for gene expression analysis, a very important thing to take into account is your research question. And one very important thing to consider is whether you want to measure the expression of one specific gene or a small set of genes, or whether you would like to perform a broader screening and measure the expression of thousands of genes and potentially even all genes that are expressed in the genome of your organism of interest. Because techniques like real-time PCR and Northern blot focus on the expression of one or only a small number of genes, whereas microarrays measure the expression of thousands of genes, and with RNA sequencing, you even measure the expression of all genes that are expressed in a certain organism. But of course, you also need to take into account practical and financial considerations, because some methods are more time-consuming than others, and also some methods are more expensive than others. So as I mentioned, I will briefly introduce the four methods that I listed to you. So we will start with Northern blot. So the first step of Northern blot is to isolate RNA from your samples. This is the case for all gene expression analysis methods. And we will come back later to how you can do this, so which methods you could use to isolate RNA. After the RNA isolation for a Northern blot, you will load the RNA onto an agarose gel and perform gel electrophoresis. So this will enable you to separate the RNA based on size. Subsequently, the RNA will be blotted, as we call it. So this means that the RNA is transferred to an RNA binding membrane, which is shown in this orange color here. And subsequently, this membrane will be hybridized with radioactively labeled nucleic acid probes. So these probes you have to design beforehand, and these will be complementary to the RNA sequence of your target gene. And subsequently, this RNA membrane will be washed to remove any non-bound probe. And then the membrane will be exposed to an X-ray film. And this generates what we call an autoradiogram. And in this figure here, you can see what this will look like. And you will see that the intensity of the band you obtain tells you something about the expression of your gene of interest. So here you can see the analysis of the expression of a gene of interest in 4 different samples. And you can see the 2 samples on the right, the expression is stronger compared to the samples on the left side. This method has several advantages, and the first one is its simplicity. And by simplicity, I mean that you don't really need a very specialized material for this. So Of course, you need to be able to perform gel electrophoresis, but this type of devices is usually available in most labs. Another interesting thing is that Northern blot will also provide information about the size of the RNA transcript that you're looking at. And this could be interesting to detect alternative splicing, for example. And also it will tell you whether your RNA product or not is degraded or not, because this is something you will also be able to see because you're using gel electrophoresis. Another advantage of Northern blot is its specificity, because the probes that you use are specifically designed to be complementary to the target sequence of interest. So therefore, the method is quite specific. Of course, Northern blot also has some important disadvantages. And the first one is that it's quite a time-consuming method, and it's also quite labor-intensive. As you see here, you need to go through several steps. So RNA isolation, the electrophoresis, the blotting, hybridization with the probes, and so on. So this will take quite some time, and it's labor-intensive as well. Another disadvantage is its relatively low sensitivity. So compared to other methods like real-time PCR, which we will discuss in detail later, sensitivity of Northern blot is relatively low, which means that when a gene is low abundant, so it's not expressed to a high extent, it will be difficult to detect using Northern blot. And another disadvantage is also that the method uses radioactive probes, which of course requires some safety precautions. And because of these reasons, in recent times, people have used Northern blot less, and we're now moving towards using real-time PCR more frequently. But of course, Northern blots are still being performed as well. So Northern blot, as well as qPCR, as I mentioned, are methods where you will measure the expression of one gene at a time. So those are mostly interesting when you want to measure the expression of only a small set of predefined genes. However, when you want to perform a broader screening, for example, when you want to test which processes are affected by a certain toxic compound, then you can use different methods. And one of those is a microarray. So here on the left, you can again see a figure of the main principle of a microarray. So what will happen is you have, in this case, two samples. One on the left here in blue is from normal cells. On the right, we have a sample derived from cancer cells. Cells are cultured, and then mRNA is isolated from these cells, and it's reverse transcribed into cDNA. This stands for complementary DNA, and we will see later what this means and how this reverse transcription specifically works. But what's important here is that the cDNA of the first sample is labeled with a green fluorescent probe. The cDNA sample from the cancer cell, in this case, is labeled with a red fluorescent probe. And subsequently, these cDNA probes are hybridized to a chip that you can see here, which contains so-called oligosequences on this microarray chip, and these oligosequences are complementary to a sequence of specific RNA targets, so specific genes of which you want to measure the expression. After this hybridization, the chip will then be scanned, and so each of these spots on a chip corresponds to one gene, and then the readout is as follows. So when the spot is gray or black, it means that the gene is not expressed in any of the cells. When it is yellow, it means that gene is expressed in both the normal cells and the cancer cells. When the spot is green, it means that gene is only expressed in normal cells. When it's red, it's only expressed in the cancer cells. And of course, the color can vary depending on differences in expression between the cell types. So with a whole range going from green to red. This is, of course, a highly simplified view because such microarray chip does not contain only 12 spots, but it actually contains 20 to 50,000 spots, which means that you can measure the expression of thousands of genes simultaneously. So that's immediately the first advantage of this method, is that it's very high throughput because you measure expression levels of 10,000s of genes at the same time. Also, this method is cost effective for a large study. So let's say you want to perform such broad screening, then of course it would not be interesting to perform real-time PCR for 20,000 different genes. Then of course a microarray would be a much more convenient option. But of course, the method also has some disadvantages. The first one is that it requires quite a large amount of RNA. So this might be difficult in some experimental setups. Another disadvantage is that the method is limited to known sequences. So in order to develop these complementary oligo sequences that are put on a chip, you need to, of course, know the sequence of your genes of interest. And this is something which we will see in the next slide. It's not required for RNA sequencing. Another disadvantage compared to RNA sequencing is that microarrays generally have a lower sensitivity, so it's more difficult to detect low abundant genes, and also small differences in gene expression between different samples will be difficult to detect using a microarray. And when you want to customize an array, so when you really want to develop a chip that will specifically allow you to measure the expression of the target genes that you're interested in, this is relatively expensive. Then another method where you can measure the expression of lots of genes simultaneously, and which is used more and more nowadays, is RNA sequencing. So in this scheme over here, you can see an overview of the principle and the different steps you would use during RNA sequencing. So what you will do, first of all, as for all the other methods as well, is isolate RNA from the samples. You will then fragment the RNA into short segments. You will convert these segments into cDNA. You will ligate these with sequencing adapters and amplify them, and then you perform next generation Sanger sequencing. The result is then that you have a lot of so-called reads, and you will then map or align these to the reference transcriptome or genome of the organism that you're working with. And then depending on the number of reads that map to a specific gene, you can then determine the expression of that specific gene in a sample and compare it between different samples. This method has several advantages. The first one is that all transcripts that are expressed in your organism are analyzed. It's also a method with a very high sensitivity and a large dynamic range, which means that it's able to detect very small quantities of transcripts, but also very large quantities of transcripts. Another advantage is that RNA sequencing is not limited to known sequences. So the reason is, of course, that here you're not working with these probes that are complementary to your genes of interest, but you're simply sequencing all transcripts that are present in your sample. And this, of course, means that when, for example, the transcriptome of your organism of interest is not fully known yet, you could still go back to the information that you obtained from RNA sequencing later on when, for example, the function of new genes has been discovered. And then you can look back at your data from an RNA sequencing experiment that you, for example, performed several years ago. And then you can still have a look at the expression of that specific gene. And another advantage of RNA sequencing is that it also allows to detect splicing variants. So you can also check whether exposure to a certain compound affects the expression of specific splicing variants of a certain gene. The main disadvantage of RNA sequencing is that it's relatively expensive, although in recent years it has become more and more affordable. And that's also why people are using it much more than in the past. And another disadvantage is that the data analysis is rather complex, so you need quite good bioinformatic knowledge, or you might need someone to assist you in the data analysis process. So those were, different methods you can use for gene expression analysis. But another one that's used a lot and that we will focus on in this lecture series is real-time PCR. So the workflow of real-time PCR is shown here. So what you will do in order to be able to perform real-time or qPCR is, first of all, you will isolate RNA from your samples. Then you will remove any residual genomic DNA. You will then convert the RNA into cDNA. And then you will be able to perform the actual qPCR reaction. Before you can do this, you first need to design primers, and you will also need to determine the efficiency of these primers. These are things we will see in the following lectures. And then you can actually measure the expression of your gene of interest using qPCR. And of course, at the end, you also have to perform data analysis and also normalize the data. And this is also something we will discuss in one of the following lectures. So we will now go through each of these steps in more detail, starting with the RNA extraction. So to isolate RNA from samples, different methods exist. And you can either use a commercially available kit that you can just purchase from specific companies, or you could also use homemade reagents. Both are possible. Of course, the commercial kits make your life easier. They can save you quite a lot of time, but of course, it's also more expensive than making all reagents yourself. But irrespective of whether you use a commercially available kit or whether you make reagents yourself, the general principle of RNA isolation is always the same. So first of all, you will collect samples and you will store them. Often samples are stored in liquid nitrogen, afterwards in -80 freezer. But when freezer is not available at that time, you could also, for example, store samples in a solution called RNA later. But the main idea of freezing the samples or storing them in the solution is to prevent RNA degradation. In contrast to DNA, which is very stable, RNA can very easily be degraded. So keeping samples at room temperature is not a good idea. Then to actually start the RNA isolation protocol, you will first lyse and homogenize the cells. You can do this either using mechanical cell lysis, and this is, for example, by grinding a sample with a mortar and pestle, or you could use some stainless steel beads and a tube, and which will then be shaken to also pulverize the sample. But on the other hand, you could also use a chemical lysis, for example, using detergents, which will disrupt cell membranes. So the idea is that you will release the components that are present inside the cells. And generally, a combination of mechanical and chemical lysis is used. In the cell lysis procedure, it's also very important to inactivate any RNases that are present. So RNases are proteins which will degrade RNA. And this is, of course, something you want to avoid. So therefore, cell lysis buffers often contain RNase inhibitors or chaotropic agents. Chaotropic agents will disrupt the 3D structure of proteins. And of course, as RNase inhibitors are also proteins, they will also be inhibited by this process. Then in the next step, generally a phase separation is performed, where solution will be divided into two phases, and RNA will be found in the aqueous phase, whereas DNA and proteins will be present in the organic phase. You can then continue to work with the aqueous phase, which contains the RNA. And then the RNA will be precipitated. Usually this is done by using a combination of alcohol and salts. Then there will be washing steps to purify the RNA, and in the end, the RNA will be resuspended. So some RNA extraction procedures, for example, those that you can buy in these commercially available kits, they work using columns. And here you can see the general principle of these column-based methods. So you have these microcentrifuge Eppendorf tubes, and this little column fits nicely in there. And at the bottom of this column, you have this silica membrane, which is used to bind the RNA. So you will first homogenize your sample, lyse the cells, and then here in this solution, these little orange stripes are the RNA. So what you will do is you will add your solution onto this column, then you will centrifuge, and then you can see that the RNA is bound to the column, and all the rest of the solution goes through by centrifugation. Then you will perform several washing steps to get rid of any organic compounds or salts that are still present. So usually there are multiple washing steps. And then in the end, you perform an elution. So this means that you just add a watery solution on top, and the RNA will come off of the column and will be collected in the solution here at the bottom of the tube. So here you can see a zoomed in image of this silica membrane. So here you can see the silica particles, and what will happen is that the salts, so the sodium cations that are present in the lysis solution, they will form a bridge between the negatively charged silica particles here and the negatively charged phosphate groups in the backbone of the RNA. And by addition of ethanol, so here in the beginning, ethanol is present, this will cause precipitation of the RNA, and this will also help the RNA to bind to this column. All the washing solutions that you use here, they will also contain ethanol, and in that way, the RNA will remain bound to the column. But in the final step here, just water is added, and this, of course, no longer contains ethanol, and as a consequence, the RNA will dissolve again and can be eluted from the column. Then another frequently used method for RNA extraction are methods based on the phenol chloroform principle. So the steps are initially the same. So you will again homogenize the sample, lyse the cells, and then you have here a solution containing all different components that are found inside the cells. So you have the RNA molecules, proteins, but also DNA molecules. You then add chloroform, which will cause a so-called phase separation. So you have an aqueous phase here at the top, which will contain the RNA, and an organic phase at the bottom, which will contain the DNA and proteins. You will then take off this aqueous phase, add isopropanol or ethanol is possible as well. This will cause the RNA to precipitate, so you get the formation of an RNA pellet. This can then be washed with additional steps of adding ethanol and removing it again. And then in the end, you can resuspend the RNA in water, and you also end up with your RNA sample. Once you've then obtained your RNA sample, it's very important to determine the RNA concentration and purity. And this is often done using a nanodrop. So this is the device that you can see here. And the nanodrop is basically a spectrophotometer. But this device, the typical spectrophotometer that you might know works with cuvettes, where you need to have a sample volume of 1 milliliter. But of course, for RNA extractions, your sample volume is usually much less. And of course, you want to not sacrifice too much of this volume just to determine concentration and purity. So the nanodrop works based on the same principle, but instead of using a 1 milliliter sample in a cuvette, you just add a drop of 1 or 2 microliters of solution onto this sample pedestal over here. Then you close this upper part, and then what you can see is that you get the formation of a liquid column. And this basically works the same way as a cuvette and a normal spectrophotometer. So what the system will do is it will send light through the sample and then measure the absorbance at different wavelengths. So specifically at 260 nanometers and also at 230 and 280 nanometers. And then the output of the nanodrop looks something like this. So here on the x-axis, you can see the different wavelengths of light, and on the y-axis, you can see the absorbance of the sample. And normally the result should look something like this. So you should see a nice peak at 260 nanometers. And the reason is that nucleic acid, so both RNA and DNA, have a maximal absorbance at 260 nanometers. So the nanodrop will then calculate two things. The first thing is the RNA concentration, and it determines this based on the absorbance at 260 nanometers via the Lambert-Beer law. This law tells you that the absorbance equals the extinction coefficient multiplied with the concentration multiplied with the optical path length. And based on this, the nanodrop software already does this calculation for you and tells you the concentration of RNA in your sample. In addition to that, the nanodrop also gives indications of the purity of the RNA. So it does this based on two ratios. The first one is the ratio between the absorbance at 260 and 280 nanometers. And if this value is smaller than 2, this indicates protein contamination. So this means that there is a high level of proteins inside your sample. This is something you want to avoid because proteins can interfere with, for example, PCR reactions more downstream in the qPCR process. And the reason is that proteins have a maximal absorbance at 280 nanometers. In addition, the nanodrop also calculates a 260 over 230 ratio. And when this value is lower than 2, this means that your sample is likely contaminated with other compounds like organic compounds, salts, or carbohydrates. So these are usually components from the lysis buffer that might be left in your sample. And again, when this value is too low, this might cause problems later on, as these components can inhibit PCR reactions. Whereas the nanodrop is used a lot to determine RNA concentration, an alternative method that can be used is the qubit. This is this device that you can see over here. And this device works with an RNA-specific fluorescent dye. And the fluorescent signal that is then emitted by the dye is proportional to the concentration of RNA. The advantage of this method compared to the nanodrop is that it has a higher accuracy. So usually the RNA concentrations determined by this device are more correct than those determined by the nanodrop. But a disadvantage is that it does not assess RNA purity. So in that case, you will not have any information about the purity of the RNA. Of course, after you perform nanodrop or qubit analysis, you will know the concentration of your RNA, which is important when generating cDNA later on. And you will also know whether your RNA is pure or not. But this is not sufficient because you also need to know whether the RNA that you isolated is still intact. Because, of course, the RNA can have a high concentration, can be very pure, so there is no contamination with proteins or other components. But when the RNA is completely degraded, this is of course also a problem. So therefore, it's important to assess the RNA integrity. And this, even though in qPCR we're looking at transcript level, so at messenger RNA, assessment of RNA integrity is based on ribosomal RNA instead, because the ribosomal RNA will also be isolated using the RNA extraction methods that we just discussed. The reason why it's based on ribosomal RNA rather than messenger RNA is that in cells, ribosomal RNA has a much higher abundance. 80 to 90% of RNA present in cells is ribosomal RNA, whereas only 1 to 5% of the total RNA is messenger RNA. And another advantage of the ribosomal RNA is that it has a very distinct band pattern, because the ribosomal RNA has very specific sizes, whereas messenger RNA is very heterogeneous in size, and it will show as a smear on when you perform gel electrophoresis. So when you perform a classical agarose gel electrophoresis on your RNA samples, this will allow you to determine the integrity of the RNA. So when the RNA is intact, and you can see an example here on the right, you will see two very clear bands with a high intensity. The upper one corresponds to the 28S ribosomal RNA, and the bottom one corresponds to the 18S ribosomal RNA. And in order to have intact RNA, the intensity ratio of these two bands should be approximately 2. So the intensity of the upper 28S band should be approximately twice as high as that of the 18S band. In case this ratio is lower, this likely means that the RNA is degraded. So for degraded RNA, what you will often see is that the ribosomal RNA bands are faint or even completely absent, as you can see in this example here. And you will also see a very strong smear of the RNA. So this is one quite convenient method to determine RNA integrity. Another method that could be used, it's actually based on the same principle, is an automated determination of RNA integrity. And this is typically done on these chips. This device that you can see here is a bioanalyzer, but there are also different devices that perform or that test for RNA integrity based on a similar principle. So what you will do here is, in each of these little wells, you can load an RNA sample. This sample has also been mixed with a fluorescent dye that will bind to RNA. Then here you will close the lid of this device. It has little needles that will send a current through the chip, and basically the small version of gel electrophoresis is also performed within this chip. And what the device will then show you is time in seconds on the x-axis. On the y-axis, you can see the fluorescence. And within this device, there is a certain detector. And you can see that at different times during the electrophoresis on the chip, you will see these peaks, which correspond to moments when a high level of fluorescence is detected. And two of these peaks correspond to the 18S ribosomal RNA fragment and the 28S ribosomal RNA fragment. And based on this pattern that you observe here, the device will calculate a so-called RIN, or RNA integrity number, and this value will range between 1 and 10. And 1 means that the RNA is fully degraded, and 10 means that the RNA is completely intact. So here you can see some examples. So the upper sample is a sample with a RIN value of 10, so you can see that it looks perfect with two peaks here for the 18S and the 28S ribosomal RNA. Here you can see at the bottom a strongly degraded sample, where you can see that the ribosomal RNA peaks are completely absent, and you have a lot of peaks here at a smaller size. And the sample here in the middle has a RIN value of 5, so it's somewhere in between. And then the device will also show you this this figure here, which shows you what this would have looked like with a classical gel electrophoresis. So you can see here a marker, so it's a letter on the left side, and then different samples. And the sample on the left has a very high RIN value of 10, so it's perfectly intact. You can also see this with two strong bands or two nicely separated bands for the 28S and 18S ribosomal RNA. And then when we move towards the right, the RIN values decrease, and you can also see this here. Sample completely at the right has a RIN value of 2.4. You can see that there is no longer, that the ribosomal RNA bands can no longer be distinguished, and you get a very strong smear here with fragments of lower size. So once you've determined the concentration and the purity and also the integrity of the RNA, you can then proceed with the next step of the procedure, which is the removal of genomic DNA. So remember that I explained that the nanodrop determines the RNA concentration based on the absorbance of the sample at 260 nanometers. But in this case, you cannot be sure that what you're measuring is only RNA, since DNA has maximum absorbance also at 260 nanometers. So the nanodrop cannot distinguish between RNA and DNA. But for real-time PCR analysis, it's very important that we specifically continue our work with RNA, and therefore we need to first remove any residual genomic DNA, because the extraction procedures for RNA will mostly isolate RNA, but of course they're not completely waterproof, so some genomic DNA might also be inside the samples. So as I mentioned, with qPCR, we should avoid amplification of genomic DNA because this will affect the accuracy of our qPCR results. We're measuring gene expression levels, so this is specifically at the mRNA level. So we want to exclude that any genomic DNA is left in the samples, as this will affect our results. So with the qPCR reaction later on, we only want to amplify cDNA, which we will generate in the next step, because cDNA is derived from our messenger RNA, and this is what we want to investigate. So to avoid amplifying genomic DNA during a qPCR reaction, we first need to remove genomic DNA. And this we can do with a DNase treatment prior to cDNA synthesis. We can do this during the RNA extraction. So especially with these methods where we use columns, we can add a DNase, so a protein that will degrade DNA. We can add this on the column, or we could also use a separate protocol after the RNA isolation is finished. And this is generally more effective, but of course it's also more time consuming because it's a whole additional protocol that you need to go through. So this DNase treatment will allow us to degrade any residual genomic DNA, but to further avoid that any genomic DNA which might still be left after this would be amplified, we can also design primers for the qPCR reaction that span or flank boundaries between two exons. I will not explain this here in detail, but I will explain it in more detail during the lecture on primer design. Once we've then removed any residual genomic DNA, we can then proceed with the cDNA synthesis. So prior to being able to perform a qPCR reaction, we need to convert our messenger RNA into cDNA, because of course we cannot perform PCR reactions on RNA. So what we will do is convert our messenger RNA into cDNA. And cDNA stands for complementary DNA, or also copy DNA. And the process of converting mRNA into cDNA is called reverse transcription. The reason is that inside cells, typically DNA is converted into mRNA, and we call this process transcription. And this is simply the opposite of transcription, so hence the name reverse transcription. The enzyme that performs this reaction is logically called reverse transcriptase. And here on the right side, you can see the principle of this reaction. So we start with our messenger RNA, or total RNA is possible as well. Then we use certain primers, and then the reverse transcription reaction is performed by the reverse transcriptase enzyme, which will produce cDNA starting from this primer. And then we end up here with our cDNA strand, and then this RNA strand, which is shown in green, will be degraded or denatured. What's very important when you perform cDNA synthesis is that you start from an equal RNA input for each sample. And to be able to do so, you need to determine RNA concentrations using the nanodrop or qubit device. So depending on the concentrations, you will dilute all samples to the same concentration in order to have the same RNA input to generate cDNA for each sample. And this is required to minimize technical variation. And this is something we will also further discuss in the lecture on data analysis. As I mentioned here, for the reverse transcription, we use certain primers. And generally, we use either oligo-DT primers or random hexamers, or a combination of both. So the oligo-DT primers, as you can see right here, they will bind to the poly-A tail of the messenger RNA, which we'll find at the 3' end of the mRNA molecule. And from there, they will allow a reverse transcription of the mRNA into cDNA. In addition to this, you can also use random hexamers. These are primers which have a length of 6 nucleotides, and they will bind at random locations in the messenger RNA, and in that way, they will ensure that all the mRNA is converted to cDNA. Because, of course, if you just use the oligo-DT primers, they will bind here on the side, but then if something goes wrong halfway through, then this half of the mRNA is not converted to cDNA. But this problem can be avoided by also using random hexamers. Then once the cDNA has been synthesized, then you can actually measure the expression of your gene of interest using qPCR. Before you do this, you first need to design primers and also test whether their efficiency is approximately 100%. But these are things we will cover in the next 2 lectures. But what we will discuss now is the principle of qPCR. So how can you measure gene expression levels using real-time PCR? So qPCR actually consists of a classical PCR reaction. And as you probably know, classical PCR reaction consists of multiple cycles, which each are comprised of 3 steps. The first step is always the denaturation that you can see here. So the 2 strands of, in this case, the cDNA will separate. In step 2, the primers will anneal, so they will bind to their complementary sequence. And these primers you have to design beforehand, and they should be specific for the gene that you want to measure. And we'll see that in the next lecture. So the primers will anneal during this step. And then in the 3rd step, the primers will elongate. And in that way, you double your amplicon, so you double your target sequence. And this will then happen in multiple cycles. So during a qPCR reaction, we usually use 40 cycles. You could even go up to 60 cycles. And then normally in each cycle, the amount of product will be doubled. Now, this is what happens in a classical PCR reaction. It's exactly the same for a qPCR reaction, but what's different is that in a qPCR reaction, we will monitor the generation of our PCR product in real time. So hence the name real-time PCR. And how can we monitor the amount of PCR product that has been formed? Well, this is something we can do by using a DNA binding fluorescent probe. And different probes exist, but the most important ones are cyber green, TOCMON, and also molecular beacons and scorpion probes. So we will briefly discuss the working mechanism or the principle behind each of these probes. So the first set of probes, which are very often used in real-time PCR analysis, are CyberGreen and EvaGreen. And these work using the same principle. So what they will do is they will intercalate between double-stranded DNA. So when they're just freely floating in a solution, they will emit low fluorescence. But once they intercalate between double-stranded DNA, their fluorescence intensity will drastically increase. So here you can see the principle. So in the first step of a PCR reaction or a PCR cycle, we have the denaturation. So you have two strands of single-stranded DNA. And you can see that the cybergreen molecules that here are shown as the green dots, they're freely floating in the solution and they emit very little fluorescence. Then the primers will anneal and they will elongate or extend. And this causes the formation of double-stranded DNA. And then you can see that the cybergreen molecules will intercalate between the two strands and they will emit fluorescence. And this fluorescence is then monitored in real time during the qPCR reaction. And EvaGreen is a similar dye, but it's relatively new and it has improved performance compared to CyberGreen. But the working principle is basically the same. The advantages of using these dyes is that they're relatively simple, so it's not very complicated to use these. They're relatively cheap compared to the other probes that we will see in a minute. And the nice thing is that they can be used to quantify the expression of any gene. So irrespective of the gene that you want to measure the expression of, you can always use the same bottle of CyberGreen. However, the main disadvantage of this specific probe is that it's non-specific. So this means that it will not just bind to the product that you want to amplify, so your amplicon of interest. But for example, when you get the formation of aspecific products, and these could be primer dimers, for example, CyberGreen will also intercalate, will also emit fluorescence, and as a result, the qPCR data that you obtain might not be reliable. So primer dimers are formed when instead of amplifying the product that you're interested in, two primers will bind to each other. This is, of course, also a form of double-stranded DNA, and the CyberGreen cannot distinguish between primer dimers or the specific product that you actually want to form. And this is, this could be a problem. Then another type of probes that are often used in qPCR analysis are so-called Tachman probes, and these are hydrolysis probes. So these are target-specific probes, so it means that they're specific for the product that you want to amplify, so they're complementary to a certain sequence of the cDNA located between the two primers that you're using. And what's specific about these probes is that they have a reporter dye shown with an R here at the 5' end and a quencher at the 3' end, which is indicated here by a Q. And then during the PCR reaction, what happens is that the probe gets cleaved by the 5' to 3' exonuclease activity of the Taq polymerase. So here what happens is you have a primer which binds here during the annealing phase of a PCR cycle. The other primer binds here to the other strand. And you also have the probe which binds here somewhere between the two primers. What will then happen is that the polymerase will start extending the primers. At some point, it encounters the probe, and then its exonuclease activity will cleave the probe. And as a consequence of that, the reporter and the quencher are separated and fluorescence is emitted. Because as the name implies, this quencher will quench the fluorescence of this reporter when they're in close proximity. But then as soon as the probe is cleaved, the two can move away from each other and fluorescence will be emitted. This method has several advantages. First of all, it's highly specific because also, like the primers, also this probe is specifically designed for the target sequence of interest. Another advantage is that it emits minimal background fluorescence. So when a probe is just freely in a solution or when it's not cleaved, it will emit very limited fluorescence. And this method also allows for multiplexing. So if you use probes that are complementary to different genes and they work with a different fluorescent molecule, you could measure the expression of several genes simultaneously. And this is, of course, not possible with CyberGreen. However, the method compared to CyberGreen also has some disadvantages. And the first one is that it's more expensive compared to CyberGreen, and also that you need to design a probe for each gene of interest. So in addition to having to design these primers, you also need to design a probe and order the probe from a company. Then another type of probes which are sometimes used in qPCR analysis are so-called molecular beacons and scorpion probes. These are also target specific. So like the Tachman probes, they also need to be designed to be specific for their target gene. And what's specific about these is that they have this hairpin shape. So you have this loop here, and then you have the two sides of, or the two ends of the probe, which will bind to each other. And similar to the Tachman probes, you have here a reporter dye on one end and a quencher dye at the other end. And as long as they're close together, the quencher quenches the fluorescence of the dye, and there is no fluorescence. What will then happen is when the probe hybridizes to the target, the dye and the quencher are separated from each other, and the dye can start emitting fluorescence. So these are so-called molecular beacons. Then the difference with scorpion probes is that the probe and the primer are incorporated into the same molecule. But the general principle of the method is the same. The advantages of this method or of these probes is that they're also highly specific because they're specifically designed for their target gene, and also that these can differentiate between highly similar sequences. However, a disadvantage is that it's more difficult to design these because they need to specifically have this hairpin structure. So these have specific requirements during design of the probe. And of course, there is also a higher cost related to those. So depending on your experimental setup, you should carefully consider which of these probes is most appropriate for the work that you want to do. Then when you perform a qPCR experiment, what the device will generate for you are so-called amplification curves. So as I mentioned, regardless of which probe you use, what will happen is that the probe starts to emit fluorescence with amplification of your specific PCR product throughout the different cycles of the PCR reaction. So what the device will do is it will monitor the fluorescence that is generated in real time. And of course, the fluorescence resembles or is linked to the amount of PCR product that is formed. So what you will get then, or what the machine will generate, is so-called amplification curves. And in these amplification curves, on the x-axis, you can see the reaction cycle. So as I mentioned, usually in a qPCR reaction, you run it for 40 cycles. And on the y-axis, you can see the fluorescence. And then you can see that these curves consist of 4 phases. First of all, you have the ground phase. So at this phase, the fluorescence is still very low, and the machine cannot distinguish it from background fluorescence levels. Then the next phase is the so-called exponential phase. And during this phase, the PCR product normally doubles with each cycle. So in each cycle of the PCR reaction, the amount of PCR product will double. Then next we have the linear phase, which is this part here. And during this phase, the reaction starts to slow down because reagents are getting depleted. Reagents could be the primers, for example, also the fluorescent probe, the nucleotides, and so on. And then in the end, you reach a plateau phase where the fluorescence is no longer increasing, and this is simply because all the reagents are completely depleted. Now, when in the end of our analysis, we want to quantify the expression of our gene of interest, and we want to compare it between different samples, we want to base this analysis on the exponential phase. So the phase that I showed you in the previous slide, where the amount of product doubles with each cycle. And here you can again see an example of an amplification curve with on the x-axis the number of cycles, on the y-axis the fluorescence intensity. And then this part here that's circled in green is this exponential phase. But as you can see, it's only a relatively short part of this figure. So what's usually done is to log transform the fluorescence. So here the values on the y-axis are put in a logarithmic scale rather than a linear scale. And what will happen as a consequence is that this exponential phase will become linear. So here the exponential phase in this little green circle is really expanded, let's say, and it gets this linear shape. And that's why we also call the exponential phase the log linear phase. So when you in the future see qPCR amplification curves, you will usually notice that the y-axis is in a logarithmic scale. Now, why is this important? Well, that is because in order to be able to quantify and compare gene expression levels between samples, we need to determine a so-called CQ value. CQ stands for quantification cycle, or it's also sometimes called a CT value, which stands for threshold cycle. So CQ or CT, it's basically the same thing. And a CQ value is the number of cycles that is required for the fluorescence to reach a predefined threshold. So this threshold in this example figure here, it's this horizontal orange line, and it's placed more or less in the middle of this exponential phase, so the log linear phase. And what you will do, or what the analysis software will do for you, is for each sample, it will determine the so-called CQ or CT value. So here you can see different amplification curves for different samples, and each of them is shown in a different color. And what the CQ value tells you is, for example, for this first sample here, this one reaches the fluorescence threshold, so this orange horizontal line, around cycle 14. Whereas for this yellow sample over here, the CQ value would be approximately 28. So this is really the number of cycles that's needed for a certain sample to reach the fluorescence threshold. And this is inversely proportional to the gene expression level in that sample. So when a sample has a low CQ value, this means that the gene is highly expressed, whereas a high CQ value will indicate a low expression of the gene. The reason is that when a gene is already highly expressed in a certain sample, you will need less cycles to reach a certain amount of product, which corresponds to a certain fluorescence. Then, as I mentioned, when you're working with cyber green, this is a very interesting fluorescent probe to work with, but the disadvantage is that it's non-specific. So in case, for example, primer dimers or other aspecific products are formed, these will also contribute to the fluorescence, and these will have an impact on the CQ value that you obtain. But in that case, of course, your data will be unreliable, because in that case, you're no longer just measuring the expression of your gene of interest, but you're also monitoring how many primer dimers are formed, for example. So when working with cybergreen, it's very important at the end of the amplification stage of qPCR reaction, you also generate a so-called dissociation curve, or also called a melting curve. So this is generated at the end of the qPCR run to verify the specificity of the product that is formed. So what will be done during so-called dissociation stage is that the temperature is gradually increased inside the PCR device. And what will happen is when the melting temperature of your specific product is reached, the DNA will denature. As a consequence, the cybergreen is released again, and its fluorescence strongly decreases. So what is then shown on a so-called melting curve is the change in fluorescence per temperature unit, and this is plotted against the temperature. So here on the x-axis, you can see the temperature, and on the y-axis, you can see the change in fluorescence per degree change in temperature. So here, this change is shown with a minus sign. So this means that when here you see a peak on this plot, it means that there is a strong drop in fluorescence. And this drop in fluorescence will happen when the melting temperature of the specific amplicon is reached. So this is what a melting curve for one sample would look like. And in this case, it seems that the melting temperature of the specific product is around 86 degrees Celsius. Now, what can also happen is that in one sample, also a second peak is found at a lower temperature. This means that that two different products are formed, one with a higher melting temperature and one with a lower melting temperature. And a lower melting temperature generally means a shorter product. And a shorter product in this case could be, for example, the presence of primer dimers. On the other hand, it's also possible that a second peak is formed with a higher melting temperature, and that could then indicate a longer product. And this might be, for example, genomic DNA, and I will come back to this in one of the next lectures. But what's very important to keep in mind is that when you see that there is an additional peak in the melting curve of a certain sample, that the CQ value that has been calculated for this sample is unreliable, and hence you should leave out or exclude this sample from your analysis. Also, here, the dissociation curve is only shown for one sample, but of course, during qPCR, the goal is to compare expression levels in multiple samples. So normally, you will see these melting curves for multiple samples simultaneously. And then it's also important to verify that the peak of these melting curves is the same for each of the samples. Of course, when you measure a different gene, in that case, the melting temperature will be different. So it really depends on the specific gene and the amplicon that you're amplifying. But in all samples where you measure the same gene, this peak in the melting curve should be at the same temperature. So now we explained the principle of qPCR. But I also want to briefly explain how this would now take place in practice. So you now know about the principle, but let's say you would go into the lab and you want to perform a qPCR experiment. How would you do this? Well, you generally would use a 96 well or even a 384 well plate, depending on the device you have available in your lab. And you will then mix in each of these wells a cDNA sample, or you could also use water. So of course you will use your cDNA samples, and then in some wells you will instead add water. These wells we call no template controls, because in these samples, or in these wells, we're not adding any samples, so we're not adding any template. And here we expect to see no amplification. In case we do see amplification in these wells, this could point towards contamination. But of course, it's also possible that primer dimers are formed, and these could also cause an increase in fluorescence. But inclusion of no template controls is important to check for contamination. And then what will you add to these cDNA samples or to the water and no template controls? Well, that's a master mix, and this master mix consists of different components. First one are gene-specific primers. So you need a forward and a reverse primer, which will be able to amplify the specific product that you're interested in. So these will be specific for your gene of interest. You will also add a fluorescent probe, for example, cyber green. Then the cyber green mix that you can buy from different companies, it will also contain nucleotides, which are, of course, important to be able to amplify the PCR product. It also contains magnesium. Magnesium is very important as it is a cofactor for the DNA polymerase, which will amplify the DNA. Then you also will add water to dilute the different components to the right concentration. And another component that's present is the so-called ROX reference dye. And this reference dye is some sort of internal normalization, which is required. And you can see the explanation of why this is necessary in this figure here. So when you have this qPCR plate, in this case, it's one with 96 wells, the device will measure the fluorescence inside each well. And differences in fluorescence will point to differences in the expression of your gene of interest between the different cDNA samples. However, the detector, which will measure the fluorescence, is closer to some wells than to others. And as a result, it will automatically measure a higher fluorescence level in the wells that are closer to the detector compared to wells that are further away from the detector. So when you measure a difference in cyber green fluorescence between different wells, it's unclear whether this is due to differences in expression level of a gene or whether it's due to differences in distance to the detector. And to correct for that, the ROX reference dye is used because this dye always emits the same amount of fluorescence, irrespective of the expression of your gene of interest. So then this fluorescence will also be higher for wells closer to the detector compared to wells further away from the detector, and then these values can be used to normalize the cyber green fluorescence. So when all these components have been mixed in your 96 or 384 well plates, you will then cover the plate with a plastic seal. And this seal is very important because, of course, the PCR reaction relies on very high temperatures for sample denaturation. And if you would not cover the plate with a seal, the samples would simply evaporate. Because I didn't include it in the slide here, but a typical reaction volume in one of these wells for a 96 well plate is only 10 microliters. So it's a very small sample volume, and this would very easily evaporate at these high temperatures. So therefore, a seal is necessary. And then once the plate has been sealed, you will also centrifuge it to make sure all solution is collected at the bottom of the wells, and then you can run the qPCR program. And the program will then consist of, for example, 40 cycles, each cycle consisting of a denaturation step, primer annealing step, and an elongation step. And then in the end, also a dissociation curve will be generated. And then once you've performed a qPCR run, you can then visually interpret the amplification and dissociation curves to check whether they look okay. And then you can export the CQ values from the analysis software. And then based on these CQ values, you can calculate relative gene expression levels using the 2 to the power of minus delta CQ method, which I will explain in detail during the session on data analysis. And then in the end, you also need to normalize your data. And this is again something we will see in the data analysis lecture.